So midterm grades are posted on the A&P website and were recorded over the weekend for the NIC grades requirements. So your midterm grades aren't in. There was a few lab grades that were missing for very few students, and so they've contacted me, and their lab instructors have given me the information to put those in. So if you're one of those students, you will see that update occurring a little bit later on. It won't occur with the midterm grades, but it will definitely be there for your final grades. So the next time you'll see a major update that includes even the lab evals will be posted on the website, and definitely it will be updated before finals are actually even in process so that everybody can see and take care of business. The one thing to remember is it was based off of 125 points, so the lowest of your 2 exam scores was dropped, because that's what we do at the end of the semester. We always drop the lowest of 6 exam scores, because we only count 5, so midterms we only count 1, and we had 3 evals, 1 notebook check, and 1 quiz. And that was because we still had some from the Thursday classes that were missing, so we couldn't put those in, and histology grades got in a little bit late. So the other thing to note, clickers were added in after the curve was calculated. We always calculate the curve without clickers because clickers truly are extra credit. And so they were added in, and that actually bumps your grade up a little bit based upon how many clickers you have. If you have been using clickers and you do not see any clicker points, that's because you have not done a proper clicker registration. I suggest you try to re-register your clicker, and if you're unsure about that, definitely stop by my office and I can see if your registration actually went through properly for you. The other thing that did not happen is deductions were not applied. Deductions will be applied at the end of the semester. So if you have any deductions, they come from where? Lab. So please contact your lab instructor if you have deductions. See if there's any way that they can be repaired. If not, realize a lot of times it's for microscope checks, or not cleaning up after yourself, or not properly logging in or documenting, signing in, things along that line. So simple things like that, make sure you're following the proper protocol so that you don't have any deductions in lab. Even though you're now dissecting on the cat, you still have to sign in, you have to do your tool checks, and you want to make sure you do a tool check before you start dissecting, and then after you are done dissecting before the class ends. So sometimes some of the instructors tell you to put a check mark when you say okay, and then when you are on your way out, put the other check mark to indicate that you've checked it twice. That's just to remind you, if you're doing it, you don't have to do that, but I would follow the protocol of your lab instructor for that. Do you guys have any questions about your midterm grades?
What was the average?
Average for the class was 69. So, and that's, as I said, without clickers. With clickers, it was 72. So that's why we calculate the curve based on things without clickers. Yeah. So clickers do help. As I said, the average was 69 without clickers, and then it went up to 72. Those of you that are not doing so well, because there's quite a few that are doing well, obviously, with that type of a curve, with that type of an average. And so you don't have to worry so much, but you also have to not be complacent, because the grade was skewed in your favor for the midterm. So those of you that did not get a midterm grade that you desired, you need to realize, do not let up. You have to continue with your memorization skills. And if what you were doing previously is not working for you, you have to change. And that's actually part of what this class teaches you. It teaches you how to become an effective learner and to learn difficult content and to change your method of studying. You can go back to the lazy way with lazy material that you have to learn, but with complicated material that you're not used to learning and retaining for any length of time, you have to come back to a more active way of learning. I just had a discussion about that with PTA program. I don't want to call him program director, because he was actually in charge of what I call the PTA program paperwork. So he's very, very, very knowledgeable about the PTA program, because he got it all set up from ground floor, and has been working with the consortium. And he said that with the IVC classrooms, in other words, with the students that are not here in the classroom when the instruction is going on, but they're in an IVC site, he says they just They go into passive learning mode, and it's terrible. It's detrimental to their learning. So that's why I always try to tell you guys, you have to learn how to become active learners. Note-taking is one of the ways to become an active learner. There are several students that do not like to take notes because it interferes with their understanding of the content that at the time that is being delivered. But ultimately, you're going to be taking notes sometime during the process because you're going to have to write some things down in order to remember them. You know, that's why we make lists when we go to the grocery store, yes? Even though you know what you need at the grocery store, if you don't make that list, are you going to forget something? So that was, you know, take a lesson from that one. You have to write it down. Even though you know what you need, you're still going to miss something, and you're going to have to go back again later on or do without. So you have to become active, and writing is an active way of doing it. Also, talking it out is an active way of doing it. Making yourself confront your fears is an active way of doing it. I'm maybe one of your fears. So it's always good to become engaged in your learning process. You know, you don't get to be a good athlete by just watching, right? So I'm asking you to become what I call intellectual athletes, because this is, even though you can't see what's going on with the brain, you have to do some things actively. Muscle memory contributes to actual learning, and that's why writing and other things are actually very beneficial to that. But you'll learn a little bit about that as we when we do the CNS chapter. We are now going back to our topic of the day, which is skeletal muscle. And realize we're using skeletal muscle as the hallmark. Remember, we use the thoracic vertebrae as kind of the stereotypical vertebrae for all of the cervical, thoracic, lumbar vertebrae in that area, because they have most of the features that we're interested in. Same thing with skeletal muscle. It has most of the features that we're interested in for cardiac muscle and smooth muscle. Smooth muscle will differ, obviously, from this because it doesn't have any striations, right? Cardiac muscle has striations that skeletal muscle has, but it's not as numerous. And we'll, when we get into the cardiac chapter, we'll learn about why that is. But that's for next semester. So I want you to realize what we're learning with skeletal muscle is going to apply across all of the muscles, the 3 different muscle types. And there will be some differences, and you should be able to actually come to and understand the various differences that are there. Remember some terminology. That's very important right now. Mesoms refer to what? Muscle and CT of muscle, right? So mesoms are referring to the CT of muscle. So we have epimesium, which is around the entire muscle. We have perimesium, which is around fascicles. And then we have endomesium. So that's all CT related. And you know the maker of CT is always fibroblasts. Some other terms. Myo is strictly muscle, and sarco. Myo and sarco also refer to muscle. So when you just have to now get the fibril, filament in line. And remember, I said one way to maybe memorize it a little bit easier is the shorter the name, the more obvious the structure. So the larger the structure. So a myofiber is a muscle cell. A myofibril are the logs or the Lincoln logs, the proteinaceous material inside the muscle cell. And the myofilaments, also called filaments, are what actually make up our Lincoln logs. And those are going to be actin and myosin. And we're going to take a look at how they interact now in more detail. The other thing we talked about was the sarcomere. And the sarcomere is a structural and functional unit of skeletal muscle. It will also be cardiac muscle, but right now we're talking about specifically skeletal muscle. And we are now going into, when we talk about sarcomeres, which go from Z disc to Z disc, and Z discs are in a light area, correct? And we call that light area I bands. And in between the light areas, obviously must be a dark area if we're talking about light areas, and we call those areas A bands. And so you have to know how the actin and myosin overlap in the I bands and the A bands, and then we also have those H zones. Realize a Z disc is the middle of an I band, and so a sarcomere is from Z disc to Z disc. So it has partial I bands on either end, and in the middle an A band. And so this is all related to actin and myosin and how they overlap in these sarcomeres. And so this is part of the striated pattern that you actually see of skeletal muscle cells. And we actually attribute this to the myofibril, because it's the myofilaments that make up the myofibril, which make up this banding pattern. It all is a little complicated at first, but the more you rehearse it, the easier it becomes to actually point out these aspects of it. When we finished last time, we were on the sarcoplasmic reticulum, because remember, sarco means muscle, and sarcoplasmic reticulum is just like, very similar, not just like, very similar to endoplasmic reticulum. It's a membranous labyrinthine sac system inside a muscle cell, and it has to surround the myofibrils. And so that's why you see this kind of odd pattern over here with the blue stuff, because the blue stuff is the sarcoplasmic reticulum. It's not at this time a requirement to understand and memorize all about the triad and the T-tubule system, but this is a system that is designed to make it more accessible from the cell membrane of a muscle cell all the way down into the various nooks and crannies inside that muscle cell with the myofibrils, because we need to actually get around those myofibrils and the myofilaments by doing so. And that's why we have that kind of odd looking system there. So realize we want to be able to get a voltage change into the innermost aspects of that cell felt. And that's part of how we actually stimulate muscle cells, is a nerve cell stimulates a muscle cell, and we're going to learn about how that nerve transmission does this. But first off, we have to understand the myophysiology of how muscle cells shorten. So this chapter now is bringing you into myophysiology, and it is not as simple as memorizing the anatomy and the basic structures. So you have to realize you have to learn the sequential steps that are now going to happen in order to understand myophysiology. And if a step doesn't happen, then the next step cannot. So this has a cascade of events that actually occur, and it's important that you memorize each step in proper sequence. With the sarcoplasmic reticulum, I drew this circle with the nucleus off to the side because it's meant to represent a cross-section through a skeletal muscle cell. And I'm going to draw my SR like a glob, okay? Not like this T-tubule system, because I'm simplistically drawing it. I just want you to understand that sarcoplasmic reticulum. And inside that sarcoplasmic reticulum is a whole bunch of calcium ions. And that's where calcium is stored. So this is a storage reservoir for calcium. And we have to get the calcium into the cytoplasm, which is also called sarcoplasm, so that it can interact with something associated with actin. Take a look at your pictures in your notes, and what is it that we want calcium to interact with that's associated with actin, that's part of the actin? Troponin. And so we want calcium to actually bind to troponin, because that binding to troponin will actually uncover the binding sites for myosin on actin. So the mere interaction of calcium with troponin actually will uncover those binding sites. So we have to get this calcium from the sarcoplasmic reticulum to those areas. In order to get to those areas, wouldn't you say it should get into the cytoplasm first? And so we need to get it into the cytoplasm. And we can get it into the cytoplasm if we put in calcium gates. So I'm going to make these little circles and make them equal calcium gates. And so I'm putting these little circles all the way around my SR. And when the gates are closed, calcium is in there. And so we need to open the gates in order for calcium to leave. And why will actually calcium leave if we open up these calcium gates? Simple diffusion. So those processes that we talked about a little while back, you have to remember these things because they drive everything. So simple diffusion, calcium will move from an area of high calcium concentration to an area of low calcium concentration. And so in order to actually open up these calcium gates, what this muscle cell will actually experience is a voltage change. So voltage delta equals voltage change will be experienced by the muscle cell's membrane. And what's another name for a muscle cell's membrane?
Sarcolemma.
Sarcolemma, that's right. So it's all these New words that we have to learn. So now the muscle cell membrane is going to experience a voltage change. The sarcolemma will experience a voltage change, and that will open calcium gates. So these calcium gates, as you can see, this SR is always, that's why this little T tubule here, that's part of the cell membrane of the muscle cell. So this sarcoplasmic reticulum is always, and you don't have to memorize the T tubule, as I said, you have to realize the sarcoplasmic reticulum is always close to the sarcolemma. And so when the muscle cell's membrane undergoes a voltage change, that is felt, by that sarcoplasmic reticulum, and that causes the opening of those calcium gates, and then calcium naturally will flood out into the sarcoplasm, because that's what it's going to do, go from an area of high concentration to an area of low concentration, and then it will be able to interact with troponin. And that's what we want to do. So that voltage change, we're going to learn, a nerve cell is going to stimulate a muscle cell, and this stimulation of, because that's what we do, we control our muscles with nerves, okay? So a nerve cell is going to stimulate a muscle cell, and that stimulus is going to then be relayed to the SR. The calcium gates are going to open, and calcium is going to flood out into the sarcoplasm and then interact with troponin. We're going to go through all of that mechanism after this. Now, once we no longer stimulate the muscle cell, the calcium gates will then close, right? So when the calcium gates close, how are we going to get the calcium back into the sarcoplasmic reticulum if it's in the cytoplasm in equal concentrations? How are we going to get that now back in? Because we want that calcium to be sequestered, so we no longer actually have that muscle cell shortening. Actively transported, and that's what we actually have. We actually have a calcium pump, a one-way calcium pump in the sarcoplasmic reticulum. See all those things that I taught you earlier? That's what you have to remember. So now we actually have a delta or triangle equaling a calcium pump. And so this calcium pump is also in the SR. And so when the calcium gates are closed, this pump is unidirectional. It only goes in one direction. And it's going to now pump the calcium back in. And when the calcium's back in, it can no longer interact with troponin, and we will no longer have a muscle contraction. So all of these mechanisms are all part of physiology. So you have to understand sarcoplasmic reticulum is a storage reservoir for calcium. It has calcium gates and calcium pumps. When the calcium gates are open, calcium leaves from an area of high concentration to an area of low concentration. When the calcium gates are closed, in order to get the calcium back inside, our calcium pumps are going. And I want you to think of these calcium pumps as like bilge pumps. Just like our sodium potassium pumps are like bilge pumps. Just for right now, maybe my story isn't exactly true, but it's true enough to get you to understand the concept. So if these calcium pumps are pumping in one direction, and they're a bilge pump, and a bilge pump is not a fast pump, right? It's designed to keep things from sinking. So the bilge pump now is able to actually put all this calcium in. Even if the calcium gates are open and the bilge pump is pumping away, isn't the calcium still going to escape as soon as you pump it back in because you're increasing the concentration? So even with the bilge pumps active, calcium still gets in the sarcoplasmic reticulum. So you can just kind of think of them always going on, happening, putting calcium in whenever they encounter it back into the SR. Now the gates are closed, calcium's in, our muscle cell is not going to contract. Now let's take a look at that contraction of the skeletal muscle fiber. Remember, that's myofiber or muscle cell. And it's called the sliding filament model of muscle contraction. And that's because the filaments, which are actin and myosin, are going to slide across one another in a specific way. Our requirements here, motor neuron, and neuron is nerve cell. So motor neuron equals nerve cell, which is different from a nerve. Must stimulate a muscle cell. In other words, zap. Okay, so I'm going to zap a muscle cell. I'm going to cause a voltage change. The muscle cell must propagate an action potential. And we like to abbreviate action potentials AP. And what this is saying that there is a voltage change that is going to be transferred to the muscle cell. Voltage change will be experienced by muscle cell. So here we are at what I was just talking about just a little bit ago. Voltage change in the sarcolemma opens the calcium gates in the SR. Calcium will flood the sarcoplasm because we want it to bind to troponin, and troponin's in the cytoplasmic area. Troponin is going to then, because calcium has combined with it or complexed with it, is going to pull tropomyosin out of the way, because tropomyosin, without calcium, is covering up the binding sites that exist on those actin little beads for the myosin heads. So troponin pulls tropomyosin out of the way. Remember, it was blocking the binding sites previously. And once we have now the binding sites to where the two can interact, this complex mechanism occurs in the following sequence. So remember, sequence is very important here. So now we are down to this part here. See the little open spots on our little beads here? Those are the binding sites on actin for the myosin heads. So they are the binding sites for the myosin heads, and that tropomyosin is no longer in the way, because that's tropomyosin right here, right? The little brown thread, so to speak. So it's no longer covering those binding sites. And again, here's that calcium that interacted with our troponin to help this process along. Now, it's really interesting. It's kind of like when you go across and you touch a live wire, you immediately feel the effect. And that's the same thing here. As soon as myosin touches actin, there is an immediate effect. And this immediate effect is what's called the power stroke. So we have to touch in order for us to have this power stroke. And what happens is, it's kind of like you reach up and you touch and then you bend. Now look at, because you're grabbing it and you bend towards your middle. Okay, because this is over here, this is one side of our myosin molecule. And the myosin molecule has all these heads on each end. Okay, and see, it bent and it pulled the actin thread towards its middle. Now, also when it did this, it's important for you to realize that there were these molecules ADP and P little I. When you see P little I, that means inorganic phosphate. So that means no carbons attached to that phosphate. But did you notice it kind of no longer has room for ADP and PI? So where ADP and PI was, whenever we actually had an immediate effect, they got knocked off. That's the easiest way to remember it. So ADP and PI got knocked off. And then we have plenty of energy, which is in the form of ATP, because we have lots of mitochondria inside these cells. And mitochondria are ATP-producing factories. And ATP will then interact with this site, which has undergone its power stroke, and it will do this in this sequence of events. We need ATP to do this. It will detach the myosin heads and reset them. That requires energy. And that's why you see in this picture over here, this resetting of the myosin head has finally used up all of our ATP. And that's what's important. ATP is required for detachment and resetting. They say cocking of the myosin head, recocking it, like cocking a gun's pistol type of thing. Trigger mechanism, excuse me. So I prefer resetting. So ATP was required for that, but it went in that sequence of events. We detached and we reset. You didn't reset while you were attached. You detached and reset. And because you are now properly aligned in this resetting, you reach up and you grab again. And you do the same thing over and over again until you run out of ATP or until calcium is removed, correct? Because when calcium is removed, what happens to the ability of the myosin heads that have been reset to interact with actin? Yeah, the tropomyosin is now blocking the binding site, and that stops the whole process. And so that's why it's important to understand when the nerve stimulates the muscle cell, it is felt by the sarcoplasmic reticulum. Calcium is released into the cytoplasm to bind with troponin to move tropomyosin out of the way. So then myosin heads can now reach up and grab, so to speak, actin and ratchet everything towards the middle of the sarcomere. That's why we actually talked about sarcomeres. And this will happen over and over again as long as A, calcium is present in the cytoplasm, and B, we have ATP, correct? And I'm just showing you with my hands just one simple myosin head, but remember when you take a look at the myosin molecules on each end, how many myosin heads are there? A lot. So what I'm showing you with one, it's happening in not necessarily parallel fashion, but in tandem so that we don't lose our grip on things and things drift apart. So that's important. And so I'm just going to do a small demonstration with my two volunteers that are going to be acting for me. I talked to them earlier before class. They told me they weren't afraid to stand up in front of people with me. So, okay. So I'm going to put you guys out here, and you guys are going to act like their bodies are Z discs, okay? And their arms out to each side are actin. Remember I told you actin is tethered to the Z disc. But the Z discs are not pounded in place. And I'm pretending I'm myosin. And so now we've had calcium in the mix. So that means I can reach up and I can grab and I can do a power stroke. And then I let go and I get reset and I do another power stroke, let go, get reset, and do another power stroke. And so, thank you, ladies. So you see that, didn't the Z disc come towards me? And so what happens to the I bands, people, whenever you contract? They get smaller, don't they? And that's part of what's important for you to realize. Did the A bands ever get any shorter when I did this process? No. So there's no shortening of our myofilaments whenever we actually contract a muscle cell or shorten a sarcomere. The shortening is of the sarcomere and of the light bands. The A bands stay the same in size. The I bands diminish because I bands are pure actin, correct? And as you pull the actin towards the center of a sarcomere, then there's more overlap of actin and myosin, and therefore your I bands become smaller and smaller to where they're almost non-existent. Because the only way that they'll be non-existent then is if myosin's touching a Z disc, right? So then you have no I bands anymore because myosin and actin are completely overlapped in that type of a scenario. But the A band doesn't change because the length of the myosin doesn't change. The length of the actin did not change. That's why it's called a sliding filament theory. Sarcomeres shorten, but no fibers shorten. So let's go ahead. You guys understand this process okay? Let's go ahead. And this is the first of your physiology. This is at the cellular level. So you need ATP for first detachment and then resetting. And that's why it says here, in the absence of ATP, you get rigor mortis. Take a look at your notes. It should say the same thing in your notes, yes? In the absence of ATP, you get rigor mortis, and we'll talk about that in just a moment. Now, I expanded my area for diagrams of sarcomeres, so you guys may have to, on your page, may have to make your make it on a different page or make your drawing at 90 degrees to your page. Because what we want to do is we want to draw some sarcomeres showing at rest. Then we want to do another one showing cross-bridge attachment. And then we want to do another one showing the power stroke. So the reason I prefer to do them in a one above the other is because I would like to draw my Z-discs. So this is my Z-disc for at rest, and I'm going to draw some actin hanging off of my Z-disc, right? And this is going to be at rest because the sarcomere doesn't shorten until the power stroke occurs, correct? Can you guys follow me with that? Because once you reach up, nothing's slid across one another. So this one is at rest. So now let's draw cross-bridge attachment before the power stroke occurs. So obviously my Z-discs need to be about the same distance apart, and I need to make my actin about the same length because nothing here shortens. Well, let's see here. There we go. Now I'm going to draw my myosin in the middle here, and because I can change the color of my pen, I'm going to do that. I'm just going to draw two myosin large molecules, and I'm going to do it here too, down below, trying to make them about the same length as the one that I have up above. And at rest, I'm going to put my little heads kind of at this little angle here, kind of like arrows, the feathers at the end of an arrow. And I'm going to put something in the way right now. What's in the way? Tropomyosin's in the way. And so we know that this is at rest because they cannot interact with one another. Now on the one down below, though, I'm going to now draw this kind of a thing. I'm going to make my quill of my arrows interact with my actin. So this is cross-bridge attachment before the power stroke. And I told you that they occur like when you touch a live electrical wire. As soon as you touch, something happens, but you have to touch first in order for something to happen. So this one here, the second one is cross-bridge attachment. And I no longer show tropomyosin in this picture because it's essentially out of the way. So if you wanted to show it still in your picture, you could put it out of the way. Like, let's see here, off to the side. You can put it off to the side. Move it out of the way. And the only reason it's moved out of the way is because calcium is present. Now, the reason I haven't drawn the power stroke is because I want you to tell me, should I make my Z-discs in the same place now as I did my previous two drawings? No. I should make them closer to the sarcomere center, correct? And I'm just going to move them over a little bit, but I want it to be done to where you can indeed understand that this is filaments sliding over one another, causing the shortening of the sarcomere. So it's kind of hard now that I got to go try and make my actin and my myosin still about the same length, but they're still supposed to be about the same length. And my myosin's going to be easy because I can just look up above and make it about the same length as I had my drawing up above. And I'm going to now do an opposite angle of my myosin heads on my actin, because I just want to reiterate, kind of reinforce the power stroke. Opposite direction than what they were before. You have to think about this when you draw this. So now there is more overlap of actin and myosin, correct? As the sarcomere shortens. No shortening of the actin, no shortening of the myosin. It's overlapping, and that's why they call it the sliding filament theory. So let's add some stuff to our notes now. Cross-bridge attachment. This happens because calcium is now available. And binds to troponin. Which pulls, because now it's got calcium with it, tropomyosin out of the way so myosin heads can now bind to actins binding sites. The power stroke occurs because of the binding. Okay? Because actin and myosin are now bound or attached, the power stroke occurs with ADP plus inorganic phosphate being dropped off. The cocking or the resetting of the myosin head occurs, this is due to ATP. First causes myosin head to detach from the actin binding sites and then resets the myosin head for further interaction with actin. Because we just have to get back into the position to jump up and do it all over again, having that ratcheting aspect. When you remove calcium, you do so by pumping it back into the SR. Then troponin no longer able to keep tropomyosin from covering the binding sites. On actin. And then if you can no longer bind to actin, what's going to happen to the filaments now? The filaments will stay the same, but the filaments will now drift apart, right? Because what's pulling, because You have to realize we're actively pulling things together to the center of the sarcomere. And when we no longer actively pull things to the center of the sarcomere, then it wants to go back to where it belongs. And so then those actin filaments, because they're attached to the Z discs, those drift back to their normal resting position. You guys following that pattern? It's an interesting pattern. And if you take a look on the OneNote site, you have a little video link here that actually is associated with this chapter and has the sliding filament model of muscle contraction, as well as actin and myosin interactions, cross-bridge cycle, and the neuromuscular junction. So you have some additional things to look at if you're having a hard time remembering things without some sort of visual reminders. So these are there for you to take a look at using this link for you. Now let's talk about rigor mortis, because we actually alluded to rigor mortis previously. Rigor mortis is because we run out of ATP, because ATP is necessary first for cross-bridge detachment and resetting of the myosin heads so that we can continue to shorten the sarcomere. So rigor mortis occurs because you're dead, right? So person is dead. But what's really interesting, your muscle cell doesn't know you're dead. That's what's really interesting with the whole aspect. So we'll talk about what that means. So person is dead. We also know ATP is needed for cross-bridge detachment. And then resetting of myosin head, if we're going to continue to use it, that is. What else do we need ATP inside that muscle cell for that we've talked about at the beginning of lecture? ATP is a source of energy for what? What did we talk about that sounds like it needs fuel? Active transport, yes. And what did we name that active transport thing? Pump. Don't pumps need fuel? Yeah. If it's a pump, do you know a pump that doesn't work off of fuel? I want to find that pump. Okay. So all pumps need fuel, right? So we also need ATP for the pumps, correct? Okay. Plus pumps. Okay. So 3 to 4 hours after you die, you start to stiffen. It typically peaks at approximately 12 hours and then dissipates at around 48 to 60 hours later. So my question to you is, why? Why this sequence of events? So when you pass away, remember the muscle cell is going to stiffen. So is stiffening kind of like a form of contracting? Absolutely. Okay. So why does that happen 3 to 4 hours later? Why do we actually have actin and myosin interacting with each other 3 to 4 hours later and then peaking at about 12 hours and then dissipating? Let's talk about why it first starts. Calcium. Calcium must be in the soup, right? In the cytoplasm of the cell. Why is calcium in the cytoplasm of the cell interacting with troponin and moving, therefore, tropomyosin out of the way. Well, the nerves are dead. You're dead. When you're dead, the nervous system's dead. But see, your muscle cells don't know you're dead yet. Because what's inside muscle cells besides actin and myosin? ATP from where? Mitochondria. What do we say about muscle cells? Do they have a lot of mitochondria? Ah. And they have something else inside them. Glycogen, yes, absolutely, and we need glycogen for that. And muscle cells, they have something important for their, no, all cells have, as long as it has a nucleus, it does have a DNA. They also have this thing that carries oxygen. What carries oxygen inside of muscles? Myoglobin. So, even though you're not circulating, you still have a source of oxygen, yes? As long as you have a source of oxygen, you have a source of ATP, right? And as long as you have a source of ATP, it will fuel your calcium pumps, right? And as long as your calcium pumps are fueled, where's your calcium? It's in the sarcoplasmic reticulum, right? Okay, so now you run out of oxygen, you run out of glucose, and therefore you run out of ATP, right? So then, because you run out of that, and now our calcium pumps aren't working, what do you suppose eventually will happen to the calcium inside the sarcoplasmic reticulum? It leaks out. Everything's leaky in life, isn't it? I don't know anything that isn't actually completely, unless it's inorganic material. Anything that's living has a little bit of natural leakiness to it. So indeed, our sarcoplasmic reticulum, the calcium will leak out, but nothing is putting it back in. And as the calcium leaks out, it interacts with troponin, which causes tropomyosin to go out of the way, which starts the stiffening process. So that's why, do you guys see how it's a gradual stiffening process? Because we have the gradual loss of calcium from the sarcoplasmic reticulum, the gradual leak out, and then we have all this calcium now interacting with troponin, moving tropomyosin out of the way, and all of those cross bridges are attached, and we've had a power stroke. And I say we've had a power stroke, because in order to have more power strokes, what do we need to have? ATP. But we already know we're out of ATP, correct? And so that's why you're stiff. Yes, you're locked in place, literally. And so then, how come you soften up after that? Why 48 to 60 hours? And this is under ordinary circumstances. Obviously, it changed with temperatures. Why do you soften up after you've been locked in place? The muscle cells are now realizing they are dead. And when you are dead, how about the stability of your own makeup? What's inside you that has digestive enzymes? Lysosomes. Everything inside you needs to be actively fueled in order to be stable. So now we no longer have ATP. Now we have calcium. Now we have interaction of actin and myosin. And now we have nothing to keep us stable. And so it's called autolysis. And isn't that what curing of meat is all about? You know, when you actually are a hunter and you kill the deer's nice and floppy at first, you know. And then it becomes harder and stiffer and stiffer and stiffer when you go home and parade it off to your friends and family. Yeah. It's a nice stiff animal, but over time it softens up because autolysis is starting to occur. And that's why you don't want to eat the meat until there's some autolysis that actually has occurred before that, because it's not going to be nice and tender. So autolysis is a form of tenderization of your meat. That's why when they kill an animal, don't they hang them in a kind of a refrigerator for days and days in order for it to become tender? It's called autolysis. So that's the rigor mortis story. So that's physiology. So now you have a taste of it, and it only gets more and more. So the first two exams, first one was purely more of a memorization focus. The second one was connect the dots. The third one is now apply physiology. We just keep adding more and more on to your abilities, because now we've actually started to exercise those neurons and get them to actually work in this fashion. It may take you a little bit more time. Some of you actually really like the physiology stuff because it's kind of a game-like thing, understanding these mechanisms. And many of you will not at first, but it's important to understand how other things work, because that's what we're actually into now, is the physiology of things. So regulation now of a contraction. So contraction is a bunch of sarcomeres shortening, because if one sarcomere shortens, isn't it next to another sarcomere, which is next to another sarcomere, which is next to another sarcomere? So when one sarcomere shortens, which is what we just talked about, they all shorten. And when they all shorten, what happens to the overall length of that muscle cell? It shortens. That's the contraction that we're actually looking at. Okay. So now let's take a look at the regulation of a contraction itself. Nerve stimulus and the neuromuscular junction. So we're going to call a neuron, which is a nerve cell that is connected or stimulates a muscle cell, a motor neuron. So this is a very specific distinction because there are different neuron to other thing connections. So motor neuron tells you it's directly going to stimulate a muscle cell. And where it stimulates it, it's called the neuromuscular junction. So it says where a muscle cell and the neuron join. So there's always going to be at least one neuromuscular junction per muscle cell. And you will also see it called motor end plate. Is another name for neuromuscular junction. Right off to the side here, this histology picture, we do not see the striations because this stain was not made to show you striations. It was designed to help us visualize the neuromuscular junction. And so this is an area where we actually have a neuromuscular junction or a motor end plate. This is another one. This one's easier to see the cell that it's attached to. And this one, you kind of get an idea that it's attached to this kind of a cell. Maybe this guy's attached to this one. This guy may be attached to this one. But this is the neuromuscular junction or the motor end plate. It's an interesting component of structures here. We have, I'm going to actually point out first, the axonal endings. So the axon of a motor neuron is coming into almost contact with a muscle cell. And so I'm going to draw this guy here. This is my axon, and this is my axonal ending right here. The axonal ending is almost coming into contact with a muscle cell, and I'm going to draw my muscle cell like this. So they are so close together that it looks like they're touching, but there is this little space between them. That's called the synaptic cleft. Tiny space between the neuron and the muscle cell, or the axonal ending in the muscle cell. Inside the axonal ending, is going to be a chemical that causes transmission or spread of an impulse from the neuron to the muscle cell. And the neurotransmitter at neuromuscular junctions is called acetylcholinesterase. So acetyl, A-C-E-T-Y-L, C-H, not an esterase, excuse me, acetylcholine. I don't know why I said acetylcholinesterase. Acetylcholine. Yeah, yeah. I still hear me when I talk. It's not just all fast forward at times. Acetylcholine, excuse me, A-C-E-T-Y-L-C-H-O-L-I-N-E. Forget the esterase part. That's coming up. So this is being dumped into the synaptic cleft, and it's going to be responsible for spreading an impulse from the nerve cell to the muscle cell. And this is a voltage change phenomenon that's going to be experienced. Inside my little axonal ending are these little synaptic vesicles, and so they're full of acetylcholine. And now we're going to actually have, in this sequence, the events of an action potential or AP propagation, which is basically the nerve cell firing and stimulating the muscle cell. So AP propagation. Okay, so you want to move a skeletal muscle. The impulse comes from the brain because that's why it says voluntary. When you see voluntary, you think skeletal muscle. That electrical impulse, which is our action potential, reaches the axonal ending, causing exocytosis of acetylcholine into the synaptic cleft. The acetylcholine is going to diffuse across the synaptic cleft, goes from an area of high concentration to an area of low concentration, and there are receptors on that muscle cell across from the, in that synaptic cleft area that are designed to bind acetylcholine. So as soon as acetylcholine's there, that will bind to that receptor, and that will then cause this step of the opening of sodium gates in the muscle cell's membrane. So there's a receptor that acetylcholine binds to, which will then stimulate the opening of sodium gates. Now, sodium is in high concentration where in relationship to the inside and the outside of a cell? Sodium is in high concentration outside the cell. What's in high concentration inside our cells? Potassium. So remember, you have to remember that sodium's high outside, potassium's high inside. And that was that sodium-potassium pump that we learned about previously. It's always putting sodium that kind of diffuses in, accidentally leaks in, back out, and the potassium that accidentally leaks out, back in. So that's what the sodium-potassium pump's all for, in addition to reestablishing voltage when it's needed. Now, sodium is an ion, and I want you to appreciate that I always draw the little pluses by things. That ion means it carries a charge, and a charge is called voltage. So if we open sodium gates associated with the sarcolemma of that muscle cell, sodium is going to flood into the cell because it's going to go from an area of high concentration into an area of low concentration. And that's going to cause a voltage change to be experienced by the muscle cell and its membrane, which, remember, that's important for the opening of the calcium gates with the sarcoplasmic reticulum. That is, as long as we are stimulating that muscle cell to fire, we are releasing acetylcholine. You guys can follow me with that? We are releasing acetylcholine as long as we want that muscle cell to contract, okay? Then when we no longer want that muscle cell to contract, we stop releasing acetylcholine into the synaptic cleft, and that is where then that enzyme, acetylcholinesterase, comes into play. This enzyme is present all the time. So even though acetylcholine is flooding that space all the time when we want to actually cause that muscle cell to contract, that means that neuron is firing, releasing acetylcholine into this cleft. Acetylcholinesterase is there trying to get rid of it. But as long as we keep flooding it, it is overwhelmed. You guys can follow me there? But then as soon as we stop flooding it, acetylcholinesterase can catch up and get rid of acetylcholine. When you get rid of acetylcholine, what will happen to the sodium gates? They close. This is the mechanism. We have to understand everything about gates opening and closing. When a gate opens, it allows something in or out, right? And so that's part of the thing that you have to understand. We have all these gates. We got to figure out what's on either side of the gate, because when a gate opens, something's going to move from an area of high concentration to an area of low concentration. And then when we close that gate, that stops that process, which will then stop other cellular processes. This is all what physiology is all about. So that's our next part here. When the action potential stops, acetylcholine is rapidly destroyed, the sodium gates close, which will then, of course, cause the calcium gates to close, which will then cause us to be able to sequester calcium back into the sarcoplasmic reticulum. So let's go to our pictures and see if we can kind of associate each picture with some of these letters up above. So our first picture right here, you guys know that this is a skeletal muscle, right? Because you see the striations on it. So that's a skeletal muscle, and this is an axon. And the little lines coming down it should be our AP, our action potential. So these guys here, events, electrical impulse, AP reaches axonal endings causing exocytosis of ACH. That's what this is all about, this picture here. It's all about B. Now let's go ahead and take a look at the magnification of B. And now we have C as this picture, yes? Because this picture is showing our little synaptic vesicles here binding to the membrane of the axonal ending and exocytosis of neurotransmitter into the synaptic cleft. So the neurotransmitter is the little kind of, I don't know whether you say blue or greenish or teal little dots there. So that's C. And then over here is D. This is showing receptors on the muscle cell binding to the acetylcholine, because here they are, here's the binding right here, binding to the acetylcholine, opening up the sodium gates, so this is D and E, opening up the sodium gates, where sodium can flood into the cell, and now we have the electrical impulse propagated from the axon of the neuron cell to the skeletal muscle cell. And so the next thing here is F down here at this bottom picture. And we see calcium being released from the sarcoplasmic reticulum here, which can now cause us to actually have actin and myosin interact. And that causes the sarcomere shortening or the contraction of the muscle cell. Once this thing happens, it's going to happen all the way. So that's one of the things that we also have to understand. You start the process and it doesn't stop just after one cycle. Because you've dumped a bunch of neurotransmitter in this area, it takes a while for everything then to undo itself once it's done. And so this will actually complete its complete cycle. In our notes here, number 6, it talks about the latent period, the period of time between the action potential reaching the synapse and the initiation of the muscle cell shortening. What's important about this is only several milliseconds. And I want to tell you what all happens during the latent period. So the period of time between the action potential reaching the synapse. So in other words, right up here, here's our action potential reaching the synapse. We have synaptic vesicles fusing with the axonal membrane causing exocytosis of neurotransmitter into the synaptic cleft. Now the neurotransmitter diffuses across the synaptic cleft. Now the neurotransmitter acetylcholine binds to its receptors on the muscle cell, causing opening of the sodium gates in the muscle cell, causing sodium to flood into the cell, causing this to cause a voltage change in the skeletal muscle cell, causing calcium gates to open up, causing calcium to flood into the sarcoplasm out of the SR into the muscle cell, causing it to interact with troponin, causing troponin to now pull tropomyosin out of the way, and causing the first power stroke. That is all during the latent period. So a lot of things are happening physiologically during that latent period. So that's an important thing to actually realize. A lot of things are happening then. Now let's get on to contraction of the whole muscle, because that was I just talked about one muscle cell right there. Now let's talk about the whole muscle organ. And that brings us to the topic of motor unit. A motor unit is a motor neuron, which is a nerve cell, plus all of the muscle cells it innervates. In other words, neuromuscular junctions occur between them. And a thing to realize, when I talk about a motor unit being turned on, it's either on or not. So a motor unit is an all or none process. So whenever a motor unit turns on, it does everything that we just talked about. And when it's not on, none of those things happen. So it's all or none. And let's take a look at two different motor units here. We'll learn about the spinal cord anatomy later on. So here we have motor unit 1, which is purple, and motor unit 2, which is kind of a reddish color. Motor unit 1 has 1, 2 neuromuscular junctions. And if you'll notice, they're with different muscle cells. Motor unit 2 has 1, 2, 3 neuromuscular junctions. If you were to stimulate motor unit 1 versus motor unit 2, if you were given the option, turning on motor unit 1 or turning on motor unit 2, which one could possibly give you a perceptible shortening of the muscle organ itself? 2. So the one that has more connections with muscle cells is more likely to give you a noticeable shortening of the muscle organ, yes? And so that's an important aspect for you to realize, because let's go down here and we're going to talk about muscle A and muscle B to make sure that you've got this. First, before we go to muscle A and muscle B, let's talk about each muscle organ possesses at least one motor nerve. And one of the things that you have to understand, a muscle is full of hundreds and thousands of muscle cells, correct? A nerve is full of hundreds to thousands of neurons or axons of neurons. Okay, so that's an important thing to also realize. Nerve equals hundreds to thousands of neurons, axons, because we're going to learn about neuroanatomy. A neuron has a single axon. So if you have a neuron, it's going to have an axon. One motor neuron may connect to anywhere from 4 to 200 muscle cells. And we want to answer this question now, knowing that which muscle, A or B, has the best control over force of contraction. Muscle A, let's take a look at it. It has 1,000 motor neurons, each attached to neuromuscular junction, a single muscle cell. Muscle B only has 1 motor neuron, and it's attached to all 1,000 muscle cells. So control over force. 1 motor neuron to 1,000 muscle cells, or a 1-to-1 connection to the muscle cells. Which can give you better control over force of contraction? A. And that's because when you have a 1-to-1 connection, you can turn on 1, 2, 3, 4, 5, 6, 7, 8 muscle cells, right? You can cause them to actually shorten until you get the desired effect. If you take 1 motor neuron, which is attached to all the muscle cells, and turn that 1 motor neuron on, that means every single muscle cell will shorten. And so if that's your biceps brachii and you want to scratch your nose, what would you do if you were built like B? You'd actually punch yourself in the nose, because you'd have every single one responding maximally. That's why I want you to understand it's all or none. It does it or it doesn't. It doesn't do it halfway. It does it or it doesn't. And so obviously one of the things that we're going to be talking about next is that in order for you to see gradations of muscle contractions, it is related to however many muscle cells are shortening. Because they're all pulling, each one's pulling on a tendon, right? Because remember those mesonems all go to make a tendon, right? And if one shortens, it's not going to have much of an effect on that tendon, right? 2, 3, 4, 5, 6, 7, 8, 9, 10. It's the however many you ask to shorten that will cause you the control over force. So indeed, we want to have our muscles more like A. They're not. They're typically anywhere from 4 to 200 muscle cells that they're actually activating or turning on. So muscle B is bloody noses. And that's what a muscle twitch is all about. This is one of your other physio X's that you guys have finished already, because it was one of your assignments before midterms. And it is the response of the whole muscle to a single electrical threshold stimulus. And threshold is that minimal stimulus necessary to actually cause something you can see and therefore record. And that's what a myogram is. It is an actual recording of a muscle twitch. It's just a recording. And it has a latent period, in other words, the time that we zapped it. The zapping takes place of the acetylcholine release into the synaptic cleft. You guys follow me? Because when acetylcholine is released in the synaptic cleft, binds to the receptors, opens the sodium gates, sodium now enters the cell. Sodium entering the cell is like an electrical current. So when we zap it, that's replacing that electrical acetylcholine sodium entry whole process. And still, we have a latent period, even with that, which you guys know is opening of the calcium gates in the sarcoplasmic reticulum, calcium diffusing out into the sarcoplasm, binding to troponin, moving tropomyosin out of the way, blah, 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 blah. All of that is occurring even in this type of latent period. So this is a latent period. We even have a latent period even with a direct electrical zap or voltage change of the muscle cell itself. Yes?
When you shiver, is your whole muscle organ responding or just different muscles?
So the question is, when you shiver, what kind of is that process? And that is actually turning off and on the contraction process, stimulating the skeletal muscles, because skeletal muscles, whenever they increase their activity, will generate some heat. And so that is a brief stimulus to actually cause contraction, and then the stimulus is gone, relaxation, and you're doing this to different muscle groups. Different motor units are doing this. And so you get that alternating muscle contraction pattern that you see with shivering. So let's take this up again. Next time we'll start here with the muscle twitch and finish this chapter.